Hello students. In this video, we'll study the histology of liver. So it is session 53 of unit 6, part of histology unit. Our body consists of different kinds of organs, and every organ has its own specific function, and functioning of all the organs are important. And when it comes to the liver, we consider liver as an important organ, one of the most important organ, because it is associated with functioning of different kinds of organ systems like digestive system and the process of digestion. It plays a role by producing bile, and it is associated with excretion process and respiration process, like this. So therefore liver is considered as one of the most important organ of our body, and functions like glycogenesis. So the excess amount of glucose which is available in the intestine or in the blood, if we consume more amount of carbohydrate, so it will be converted into glycogen. So that process takes place in the liver, and glycogen is stored in the liver itself. Whereas the vitamins, so excess amount of vitamins are stored in the liver, and liver is involved in the production of bile juice. So this bile juice plays a very important role in the digestion of lipids, and the synthesis of urea, the process or the cycle called ornithine cycle, where the ammonia is converted into urea, that also takes place in the liver. And detoxification, so any harmful molecule or chemical which enters into our body, that is taken care by the liver. Say for example, a person who chews or smokes tobacco, the nicotine enters into the blood, and that nicotine is broken down by the liver. So like this, any medication, any harmful chemical that enters into our body, that is taken care by the liver by means of detoxification. So probably this is one of the very important process or functioning of the liver. And it is a very large sized organ, and it weighs about 1.5 kgs in a human. Now when it comes to the histology of liver, there is a structure called Glissen's capsule. The liver is covered by a fibrous capsule, and that is called Glissen's capsule. It actually covers the whole liver, and the base of the liver is called hilum, and this Glisson capsule covers the hilum also. And in the section we can see the hilum consisting of the portal vein, hepatic duct, and hepatic artery. So all these vessels are covered by Glisson's capsule, and Glisson's capsule actually continues to cover the lobes of the liver. And when we observe the organ of liver, we find that liver is divided into four lobes. So two larger lobes and two smaller lobes. So the large ones are left and right lobe, and the two smaller lobes are commonly called quadrate and caudate lobes. So like this, we can say the liver is made up of four lobes, two larger lobes and two smaller lobes. Under the section of the liver, uh, under the microscope, shows following details. We find hepatic lobules. Uh, Parenchyma is the functional tissue of the liver, and it is organized into the hepatic lobules. So the cellular part of the liver is commonly called the parenchyma, and that is the functional tissue. So whatever the functions, whatever the function we attribute to the liver, so those functions are performed by parenchyma cells. And these parenchyma cells are actually organized into structures called hepatic lobules. So you can say hepatic lobules represent the units of liver, and these hepatic lobules, of course, they are made up of liver cells, and we commonly call it as parenchyma. And you can see This hepatic lobule is polyhedral in shape. It appears like a polyhedral shape in section, and it shows some uh, parts like central vein at the center, and we find the hepatic arteries and hepatic portal veins supplying blood. In detail, we can study about hepatic lobule in the next slide. And large number of hepatic lobules are found in liver. So structurally, liver is made up of very large number of such hepatic lobules. So you can imagine the hepatic lobules uh, present in a very compactly arranged manner, and that together forms the liver. So liver is simply packed with structures called hepatic lobules, and these hepatic lobules have dual blood supply. So liver is actually supplied with two blood supplies. So that is from the terminal branches of hepatic portal vein and hepatic artery. And we know the portal systems. In our body, we find these portal systems into kidney and in the liver. Okay? Probably you know what are portal systems and what is the significance of those portal veins. So liver is provided with portal veins and hepatic artery. Then sinusoids are the structure through which the blood flows between the hepatocytes, and this blood then drains into the hepatic lobules, central vein. So actually in the hepatic lobule we can see the cells, and in between the cells we find some spaces, and those spaces are called sinusoids, and the blood actually flows through those sinusoids and ultimately it reaches the central part called central vein. Now in this diagram, let us see it in detail. So this is at a lower magnification. You can see here this is one lobe, hepatic lobule, right? So this hexagonal shape represents one hepatic lobule under the microscope. So this is how it appears under microscope. This is a transverse section of the liver tissue, liver. And in higher magnification, you can see here this is the central vein. So in a hepatic lobule, there is a central vein, and these are the liver cells. And the central vein is actually a cavity into which the blood drains at the end. So you can see the spaces in between the cells, and these spaces commonly form the sinusoids. Right? Through these sinusoids, the blood actually flows. So from the edges, here we find the blood vessels, and the structures which are found here are called triads. So we'll study about triads in the next slides. So the triad structure consists of blood vessels and also the bile duct. So the blood which comes from here, it will pass through the sinusoids which is present between the cells, and ultimately it will drain into the central vein. So this is how the structural arrangement of the liver can be understood. So hepatic lobule, and here you can see the central vein and the space between the cells, the space which forms is called sinusoid, and at the junction of hepatic lobules we find structures called hepatic triads, where we find bile ducts, blood vessels, lymph vessels, etc. Now let us try to understand hepatocytes. Hepatocytes are nothing but liver cells, the structural and functional part of the liver. So parenchyma cells is a general term, and specifically you can call them as hepatocytes. So hepatocytes of liver or liver cells are the functional part of the liver. So in this picture you can see a magnified view of a hepatic lobule, and this is the central vein, and you can see the hepatic cells or hepatocytes. Actually what happens is this is the central vein, and opposite direction you find the periphery of the hepatic lobule. So the cells are actually arranged in the form of cords, and we call them as hepatic cords. So the hepatic cords actually extend from the central vein to the periphery. So this is how the arrangement can be seen. So these are the cells, and in higher magnification you can see the liver cells looking like this. And one important thing you have to remember is liver cells are one of the most active cells of our body because they are involved in so many different kinds of functions. So a lot of biochemical activities happening inside the cell, and therefore as a source of energy they need a lot of ATPs, and therefore we find very high number of mitochondria in the liver cells. So presence of more number of mitochondria is an indication of activeness of the cells. Now these cells are known to perform many kinds of metabolic activities like production of bile. So we already discussed about this. So the functions of liver are performed by liver cells itself. So the functions include production of bile, and number of plasma proteins, so the proteins, and non-essential amino acids. Now this is probably a very important function. Plasma proteins means the proteins which are part of the plasma of the blood, and amino acids, we know there are two kinds of amino acids, essential and non-essential amino acids. So essential amino acids will be available to the body through the diet. So the amino acids which are going to be produced by the liver, we need not take it in the form of food. So liver also has a function of production of amino acids, and it is also involved in metabolism of fat, metabolism of carbohydrate, and metabolism of proteins. Okay, and as we already discussed, storage of glucose. Actually, it converts the glucose into glycogen and then it stores glycogen. Storage of vitamins, storage of minerals like iron, and the most important function, not most important, but one of the very specific function is detoxification of metabolic waste products and toxic materials. So that is about the hepatocytes. And let us continue our study of histological details of the liver. So a hepatic lobule at lower magnification, the hexagonal shape of the hepatic lobules can be identified by their slightly darker edges. So under lower magnification, if you observe the transverse section of a liver under the microscope, you can identify the lobules very clearly. How? You can see here, this is one lobule and this is another lobule. So you can see the lobules. We can differentiate lobules by the dark borders with which they get separated from each other. And at the center, we can see the central vein very clearly. So one hepatic lobule has one central vein. So in each hepatic lobule, you can see one central vein like this. So this is how under the microscope the section shows hepatic lobules very clearly, and the structures called portal triads or hepatic triads are found at the junction of hepatic lobule. So you can say this is one junction and this is another junction, and these are the places which are called hepatic triads, where we find the bile ducts, portal veins, and arteriole. So all these structures like lymph vessels, blood vessels, nerve endings, all these structures are actually found in the portal triads or hepatic triads. Now, okay, this is just the magnified view of one hepatic lobule, where we can see the central vein, and this is the hepatic triad. So you can clearly see the hepatic triad showing portal vein, which is a large vessel, I mean comparatively it is having larger diameter, and hepatic artery which is smaller in size, and the bile duct. Now this area is called portal area, or this structure is called portal triad or hepatic triad, which consists of portal vein, hepatic artery and bile duct, and this is the lobule. So the blood actually comes from this artery and vein, and it drains into the central vein. Now let us move on to understanding of hepatic triads or portal triads in detail. So portal triads are typically located at the corners of the lobules. We already discussed about it. The portal triads consist of large thin walled portal vein. So veins are having larger diameter and they are thin walled, whereas the arteriole is smaller in size and it is thick walled. So this is the vein and this is the artery. This is larger in size and this is smaller in size. And the one more structure we find is the section of bile duct. Now we can easily differentiate the artery and bile duct because bile duct is having typical cuboidal epithelium. So the presence of cuboidal epithelium uh, indicates the bile duct, whereas the arteries are lined with squamous epithelium. Now hepatocytes. We already discussed about hepatocytes. Hepatocytes are large polygonal epithelial cells that form branching plates that are only one cell thick, separated by sinusoids and radiate outward from the central vein. So I already told you hepatic cells join together to form a hepatic cord, and these hepatic cords actually extend from the center. So what is in the center? Central vein. So from the central vein they extend up to the border of the lobule or the periphery of the lobule. So that is how the hepatocytes are organized, and chains of hepatocytes are commonly called hepatic cords. Now the sinusoids, now this is what we have to understand. Sinusoids carry blood from the hepatic arteriole and portal venule to the central vein, while the bile canaliculi or capillaries carry the bile produced by the hepatocytes in the opposite direction in order to drain into the bile ductules. And hepatocytes contain large number of mitochondria. Oh, that we already discussed. Now what is this point? So sinusoids carry blood from the hepatic arteriole and portal venule to the central vein. So we already are familiar with this structure. So this is the hepatic lobule, and here are the hepatic triads. So this is a diagrammatic representation of the hepatic lobule. And in the hepatic triad, what we find? We find vein, artery and bile duct. Now vein and artery, they contain blood. So the blood actually flows from the artery and vein towards the central vein. So this is the direction of blood flow. So through the sinusoid the blood flows. So this is the direction from triads to the central vein. Now these cells, the hepatocytes, they have many functions, and one of the function of hepatocytes is secretion of bile juice. So bile is produced by hepatocytes. So the bile which is produced by the or secreted by the hepatocytes, it actually moves in the opposite direction. So the bile juice will move from the hepatocytes or the lobule, hepatic lobule, towards the hepatic triad, right? So the blood flows from hepatic triad to the central vein, whereas the bile will flow from not from the central vein but from the the hepatocytes towards the hepatic triad, because it is the bile duct which is going to carry the bile juice, and bile duct will ultimately open into the gallbladder, which will open into the duodenum of the alimentary canal. And finally, we can summarize that liver is the largest organ found in the body, and it's covered by Glison's capsule. And liver, histologically, liver shows hepatic lobules, and these hepatic lobules are made of the functional and structural part of the liver, that is hepatic cells or hepatocytes or liver cells. So all the functions of the liver are actually performed by the liver cells. Now that is the study of histology of liver. Thank you.